Welcome to Washington America Show. The real prospect of effective vaccines being available in a matter of months renders every coronavirus death even more of an avoidable tragedy than has been the case from the beginning of the pandemic. The way forward is to socially distance, clear the air, wash hands, and mask up until a vaccine opens up a controlled, safe road to herd immunity. The efficacious vaccine is going to be essential to prevent further mortality. In this unprecedented pandemic, vaccine development is time-dependent, and considerable collaborative efforts are being expended to expedite preclinical and clinical assessment of candidate vaccines. The cost to manufacture and deploy an efficacious COVID-19 vaccine will be vast. The novel coronavirus, severe acute respiratory syndrome coronavirus, has caused more than 1 million deaths in the first 6 months of the pandemic and economic and social upheaval. An efficacious vaccine is essential to prevent further mortality. Although some countries might deploy COVID-19 vaccines on the strength of safety and immunogenicity data alone, the goal of vaccine development is to gain direct evidence of vaccine efficacy in protecting humans against SARS-CoV-2 infection and COVID-19 so that manufacture of efficacious vaccines can be selectively upscaled.
A candidate vaccine against SARS-CoV-2 might act against infection, disease, or transmission, and a vaccine capable of reducing any of these elements could contribute to disease control. The most important efficacy endpoint, protection against severe disease and death, is difficult to assess in Phase 3 clinical trials. In this review, we explore the challenges in assessing the efficacy of candidate SARS-CoV-2 vaccines, discuss the caveats needed to interpret reported efficacy endpoints, and provide insight into answering the question, does this COVID-19 vaccine work? Keep watching this video.
Although some countries might deploy COVID-19 vaccines on the strength of safety and immunogenicity data alone, the goal of vaccine development is to gain direct evidence of vaccine efficacy in protecting humans against SARS-CoV-2 infection and COVID-19. In their target product profile for COVID-19 vaccines, WHO suggested that a clear demonstration of efficacy, on a population basis, ideally with 50% point estimate should be a minimum criterion for any acceptable COVID-19 vaccine, and that efficacy can be assessed against disease, severe disease, and/or shedding or transmission endpoints. This definition is nonspecific and reflects the complexities of assessing the clinical efficacy of candidate vaccines in the context of a novel pathogen.
A COVID-19 vaccine capable of reducing any of these elements might contribute to disease control where there are no efficacious prophylactic medications and few treatments. The US FDA suggested that laboratory-confirmed COVID-19 or SARS-CoV-2 infection are appropriate primary endpoints for vaccine efficacy studies, with an endpoint estimate of at least 50% for placebo-controlled efficacy trials. Protection against severe disease and death is difficult to assess in Phase 3 clinical trials due to the unfeasibly large numbers of participants required. Instead, data to address this endpoint might be available only from large Phase 4 trials or epidemiological studies done after widespread deployment of a vaccine. It is possible that alternative vaccine platforms are required for adequate immunogenicity in older age groups.
Prospective studies of vaccine effectiveness in real-world scenarios post-licensure are routinely needed. In the case of SARS-CoV-2, an efficacious vaccine might prevent infection, disease, or transmission. The outcome of SARS-CoV-2 infection in individuals is heterogeneous and dependent on multiple variables, including age, sex, ethnicity, and comorbidities. On an individual level, the consequence of infection can range from post-symptomatic states to hospital admission, requirement for respiratory support, and death.
The ability of a vaccine to protect against severe disease and mortality is the most important efficacy endpoint, as hospital and critical care admissions place the greatest burden on healthcare systems. The beneficial effects of such a vaccine on a population can be observed only if the vaccine is efficacious in older adults, approximately greater than 60 years, and widespread distribution of the vaccine exists, including to people who are most susceptible to COVID-19.
High coverage among these groups who are at high risk of severe COVID-19 would have the greatest effect against disease endpoints. Alternatively, vaccines that do not affect the clinical course, but reduce the transmissibility of SARS-CoV-2, could still be valuable interventions on a population level. Although vaccine candidates can be assessed in isolation, WHO and the US FDA suggest that an adaptive trial design, evaluating multiple vaccine candidates in parallel against a single placebo group, could be an acceptable method to increase efficiency, provided that the trials are sufficiently powered.
Studies that rely on natural exposure to SARS-CoV-2 are vulnerable to multiple variables that influence whether a vaccine is exposed to SARS-CoV-2 and then whether exposure leads to infection. For example, older participants could be more likely to avoid social gatherings, reducing their likelihood of exposure to SARS-CoV-2. However, healthcare workers might not only be more likely to be exposed to SARS-CoV-2 but also might receive higher infecting doses than would other participants in the study.
By virtue of their recognized high-risk occupations, healthcare workers might have better access to protective strategies than other participants, reducing the likelihood of infection following exposure. These complex behavioral variables are difficult to control, so it is important that participants are randomly assigned between vaccine and comparator groups, by use of a concealed method, to ensure reliable assessment of efficacy outcomes.
Efficacy studies should be adequately powered to meet efficacy endpoints, and multiple variables inform these calculations, including local transmission rates and participant characteristics. For example, the severity of COVID-19 and mortality rates vary according to age, sex, and ethnicity. If a Phase 3 efficacy study enrolled participants aged only 20 to 29 years, the expected low mortality rate in this population would require an unfeasibly large sample size to adequately power the study to assess mortality as an endpoint.
Given that older participants, especially people with comorbidities, are more likely to socially shield, they might be less likely to be exposed to SARS-CoV-2, and so a mortality efficacy endpoint might still not be met. Recruitment of older participants in vaccine trials has been historically challenging. Dedicated trials will be needed to assess COVID-19 vaccines in individuals younger than 18 years, as trial data in adults might not be predictive of vaccine safety and efficacy in this age group. Since children are less affected by COVID-19 disease than are adults, substantial safety data should be collected from adults, and greater understanding should be acquired of the biology of pediatric multisystem inflammatory syndrome temporally associated with SARS-CoV-2 infection, before pediatric vaccine studies are initiated. Incidence of SARS-CoV-2 infection varies, with hotspots and surges occurring in an unpredictable way. This unpredictability has a considerable effect on study sample sizes for efficacy endpoints, especially as infection rates are highly likely to change over the study follow-up period. Wear a mask, save lives. Wear a face cover, wash your hands, keep a safe distance, cover your cough or sneeze with a tissue, then throw the tissue in trash. Wear a mask, save lives. Wear a face cover, wash your hands, keep a safe distance, cover your cough or sneeze with a tissue.
Wear a mask, save lives. Wear a face cover, wash your hands, keep a safe distance, cover your cough or sneeze with a tissue, then throw the tissue in trash. Wear a mask, save lives. Wear a face cover, wash your hands, keep a safe distance, cover your cough or sneeze with a tissue, then throw the tissue in trash.
To allow comparison of efficacy between vaccine candidates and within differing populations, it is essential that standardized, quantifiable endpoints are applied routinely to clinical trials of COVID-19 vaccines, and that their limitations and potential for bias are understood. The COVID-19 Clinical Working Group of the Coalition for Epidemic Preparedness Innovations has published guidance suggesting that the primary endpoint for assessment of efficacy should be virologically confirmed COVID-19. As understanding of COVID-19 grows, so does appreciation of the heterogeneity of the symptoms and signs associated with SARS-CoV-2 infection. The clinical criteria chosen to prompt diagnostic testing needs to carefully balance sensitivity, to ensure all cases are identified, with specificity. Wear a mask, save lives. Wear a face cover. Wash your hands. Keep a safe distance. Cover your cough or sneeze with a tissue, then throw the tissue in trash. Wear a mask, save lives. Wear a face cover. Wash your hands. Keep a safe distance. Cover your cough or sneeze with a tissue, then throw the tissue in trash. Wear a mask, save lives. Wear a face cover. Wash your hands. Keep a safe distance. Cover your cough or sneeze with a tissue, then throw the tissue in trash. Wear a mask, save lives. Wear a face cover. Wash your hands. Keep a safe distance. Cover your cough or sneeze with a tissue, then throw the tissue in trash. Wear a mask, save lives. Wear a face cover. Wash your hands. Keep a safe distance. Cover your cough or sneeze with a tissue, then throw the tissue in trash.
Please share this video, subscribe to our channel.